Good morning, everyone. In this module, we are going to discuss about gene regulation with the help of an example, lycopene. So before that, we will discuss what is gene regulation. So we know in human beings, there are approximately 30,000 genes, and each gene will function in different times and in different stages of that particular period. So how this gene regulation is taking place? So when we discuss, the genes are located within the DNA inside the nucleus. So from that, the genes are transcribed in the form of mRNA with the help of process. That process is called as transcription. So the message from the DNA is transcribed in the form of codonic sequences in mRNA. So the gene regulation can be, the expression of gene can be regulated in the transcription level. And from the nucleus, once the mRNA is formed, then the mRNA can be transported to the cytoplasm. So before transporting, the mRNA will be, which is primitive form or primary form of mRNA is hnRNA. Then from hnRNA, it is matured in the form of mRNA. So there can be a gene regulation or a regulation or a control at the processing step. That means the conversion of hnRNA to mRNA with the process of splicing. Then the third level of regulation we can observe in transport of mRNA, the matured form of mRNA from nucleus to the cytoplasm. So it can be regulated in that transporting region. Then the final one, that is the actual protein synthesis, the product of gene or the expression of gene resulted in the form of protein within the cytoplasm with the help of ribosomes and tRNA, mRNA, etc. So that process we'll call it as translation. So even in the translation step also, the regulation can be possible. So once again, the regulation, the gene, whether the gene is to be expressed in such a way, in that exact form, and it can be regulated on the ongoing process of protein synthesis. So it can be the primitive step or the first step itself, it can be regulated in the transcriptional level. That only we are going to deal in this topic. Okay, how the genes are regulated in the transcriptional stage. And not only in the transcriptional stage, it can be regulated in the processing level. That means maturation of hnRNA to mRNA. Then it can be transport of mRNA to the cytoplasm. That means to the site of translation, that is in the ribosomes. And even at the time of translation, that means at the time of protein synthesis also, gene regulation can be controlled. But in this module, we are dealing with the transcriptional level of gene regulation. So when we are discussing, before discussing that one, we have to deal with the types of genes. So there are two types of genes. The first type, it is called as constitutive genes. That type of gene, it is called as luxury genes or non-constitutive genes. So this, with the help of an example in our day-to-day life, we can explain, because the genes are regulated or genes are expressed in the form of protein. So the same way, if we are comparing to an example, in our house, there are many electrical equipments. Fan is there, fridge is there, washing machine is there. Then sometimes AC will be there. So different electrical appliances are working in different stages and different seasons and different situations, etc. But each of these activities of this electronic equipment is regulated with the help of switches or switchboard itself, we can say. Whenever we need a fan during the summer days, we can switch on that one and we can avail the facility or working of that one. That functioning can be utilized. The same way, the washing machine. Whenever we need to wash our dress, we can function that washing machine switch on. Other times it can be made it off to conserve the energy. The same way itself in our body also. The requirement of proteins. So when we are saying the proteins, it can be hormone. Say for example, insulin. When the food reaches into the stomach, into the digestive system, that time only into the small intestine we can say exactly. When it reaches the small intestine, that time only insulin has to be secreted and the glucose level has to be regulated. Otherwise, there is no need of insulin in that near vicinity. And when the child is growing period, that time only we need the growth hormone. So like that, each and every protein or hormone or whatever it is. But here we are discussing about protein. That's what I said. The insulin or growth hormone also more or less the same. During the child uh, developmental ages, that time only growth hormone is required more than the normal cases. So like that, each and or during the lactation period only, the milk protein has to be, lactalbumin has to be produced. So which one, according to the necessity, according to the need, that time only that particular protein can be muscular protein, collagen or elastin, or it can be hormones, say for example insulin, or it can be any other substances, regulators or antibodies. When your body is affected with the diseases, that time only we have to produce immunoglobulin. So in that way, according to the need, that time only that particular type of protein has to be produced, even though that protein production is controlled and their gene is located in the DNA within the nucleus of all the cells. Certain necessities or at requirement time only that gene has to be expressed. So this expression of the gene in the form of proteins is regulated with the help of certain regulatory mechanisms. Collectively, we can call it as the gene regulatory mechanism. So the same example of our household activities, I said, there are certain genes or there are certain electrical equipments which has to be on throughout. A common example if you are taking means if you are using fridge in your house, that power plug should be always on. Then only the food items which are kept within or which are stored within the freezer will be maintained in that particular temperature. Every now and then, if you are switching it and switching on and off means the functioning won't be proper. So there is a continuous supply of switching on or on condition should be maintained in order to function that particular equipment in a proper functioning way. Instead of that, if it is AC means whenever the outside or whenever you feel that the room should be cooled, that time only it has to be on, otherwise it has to be off. Because in order to conserve the electricity, consumption of the electricity, we are regulating it with the help of a switch, either it on or in off condition. The same mechanism is adopted within our body also. There are certain enzymes or certain proteins which has to be produced throughout without any regulation. Its presence is compulsory throughout in order to maintain the life. Then that type of proteins or that type of gene regulation itself, we can call it as the constitutive genes. That means that genes will be expressed always, and as a result, that type of proteins will be produced always. So if we are saying any example means the glycolytic enzymes. So we need the production of energy or Krebs cycle enzymes which are involved in glycolysis and enzymes which are involved in Krebs cycle. Here, these enzymes are essential because from that, with the help of that enzymes only, glucose will be, first step, it is converted into pyruvic acid. So there the glycolytic enzymes are compulsorily, their presence is essential. And the same way the pyruvic acid will have to be converted into carbon dioxide and water and then ATP. Because in order to maintain the life in an organism, a continuous supply of ATP is needed. So these two enzymes are throughout their presences or their production is essential. So that means if these enzymes have to be produced means this genes which regulate the production of these enzymes. Glycolytic enzymes as well as Krebs cycle enzyme should be always in on condition, and such type of genes which are always expressed in the form of proteins, they are called as constitutive genes. So they always will be in on condition, always their mRNA will be produced, and from that mRNA always the translation will take place. So transcription also will take place, translation also will take place, and the polypeptide synthesis will take place, and their presence is ensured throughout in order to maintain the life of the organism. There are another set of genes that are called as luxury genes or non-constitutive genes. Whenever that need arises, or whenever that presence of that enzyme or any protein molecule is required, that time alone is that molecule is produced. Nowadays we are facing with corona. So whenever any corona outbreak or any coronavirus entry, that time only our body should produce the antibodies against the coronavirus or any other pathogen. Just for an example, I have quoted the corona. So whenever any disease pathogen is entering, that time to fight with them, our body will produce antibodies. So smallpox we are not at all nowadays encountering, so our body is not producing any antibodies against the smallpox. But when that situation, if any like that emergency situation arises, that time automatically the genes which are present in the DNA will be in on condition, will transcribe in the form of mRNA. Then from the mRNA, translation will take place in the form of protein or polypeptide. Then that proteins only will be modified either in the form of antibodies or in the form of hormones or in the form of any other structural protein, so and so. So that type of, so there is an essentiality of regulation, at what time it should be on and when it, it has to be made off. So this gene regulation only we are going to discuss more in this one, how the non-constitutive pattern of gene regulation is taking place. And there are the typical examples. So in that way, we can arrange the switching on and off of the gene, and in that way, the protein production can be regulated. So here, this concept was, the gene regulation, that concept was proposed by two scientists, Jacob and Monod. And they named that one gene regulation in the transcriptional level, that is called as, that is named as, that process is gene regulation and the regulation of protein synthesis at the transcriptional level was proposed by the scientists Jacob and Monod, and they called that gene regulatory unit which regulates the expression of a gene, that is named as operon. So operon is a group of genes that are transcribed at the same time, and they usually control an important biochemical process. We can say any metabolic process. And the typical example they have quoted is the lac operon. So lac operon means lac stands for lactose. So they have taken the example from the lactose utilization of E. coli, which is a prokaryote, Escherichia coli. So E. coli, as we know, it is a bacteria. So they need a nutrient medium for growing and multiplying and enlarging their colony. So normally, in the normal cases, the E. coli will utilize which one? The glucose as their source of energy, source of carbon, we can say. So whenever the glucose is there in the immediate surrounding, that means mainly E. coli can be grown in agar medium. So when we add in the agar medium the glucose, normally the E. coli will utilize the glucose and they can derive the energy from the glucose metabolism. But suppose if the glucose supply is stopped, then in order to survive them, if you are adding lactose, you know, glucose is a monosaccharide and lactose is a disaccharide. So they can survive even in the medium which contain lactose in the absence of glucose. So that means they require, that is the source of carbon. So in order to utilize it, they need the presence of certain enzymes. And the main enzyme they need is beta galactosidase. With the help of beta galactosidase, the lactose can be broken down into glucose and galactose. So this metabolic process that can take place, or that the enzyme production which mediates the glucose lactose utilization, can be regulated with the help of a regulatory system that is called as lac operon. So here the lac operon, with the help of lac operon, the E. coli, even though the absence of glucose, and into that medium, if you are adding lactose, they can survive in that lactose medium, because at that particular moment, in the absence of glucose, the presence of lactose, they will produce a set of, with the help of gene regulation or switching on of a gene regulatory unit. And in that presence of on condition, the transcription will take place and a group of enzymes will be produced. That enzyme will act on lactose and convert this one into glucose and galactose. So the end product is the glucose, so they can easily utilize the glucose and they can survive in that lactose. Then the same way there are, so this operon means it is a regulatory unit. It will contain a sequence of, or a series of, or a group of genes. In that gene, we know already that I said there is, it regulates the transcription, the production of enzyme, the production of protein with the help of an mRNA, transcription. So for that transcription to take place from DNA to mRNA, they're essentially, or it is essential that they should contain a promoter, then they should contain structural genes, and apart from that, there are many other sets of genes. As such, we'll call it as the lac operon unit. So it is, this type of gene regulation is found not only in E. coli, there are many other metabolic pathway also operons are functioning. So a few examples that is quoted, that is lac operon, that is in, it is all in E. coli, tryptophan operon, arabidose operon, then histidine operon, valine operon. So there are many different types of gene regulation. As an example, nowadays it is studied very in a detailed manner. And for us, we have to learn only lac operon. So here, this is the pictorial form of the gene regulation with the example lac operon. So here in this first one, we are showing a gene sequence or a DNA sequence. In that DNA sequence, we can see many genic unit. So the first one, it is the more first and foremost, or the components of lac operon. When you are saying there is an I gene, so just left to that I gene, there is a P, that is a promoter unit or promoter region. Promoter means we already know the region where the RNA polymerase binds and takes initiation of the transcription. So here this is an I gene, and I gene, it is I stands for inhibitor. I stands for inhibitor, and that gene is constitutive. So here already we have to recall the difference between constitutive and as well as the difference between the luxury gene. So IG means it is always expressed. There is no, even though there is a promoter, the promoter will be always on, and that promoter will allow its own RNA polymerase to bind, and as a result, the I gene will transcribe into repressor mRNA, which is a protein. And this repressor mRNA can regulate the expression of that gene. So how it can regulate that, I have to explain the remaining components of the lac operon. So there is an I gene, which is not near to the operon region, but slightly far away, little more far away from the promoter and the structural gene regulation unit. It is slightly far away. It is having its own promoter site, but that promoter always will allow the binding of its own mRNA repressor RNA polymerase to this one. So always constitutively the repressor mRNA will be transcribed, and as a result the repressor protein will be produced. Then the next regulatory unit, one of the component is promoter itself. Again here also another promoter, that only more important. That second P allows the binding of RNA polymerase for the regulation, for the transcription of mRNA of lac operon. It will allow the or it will takes initiative for the regulation or for the transcription of the genic transcription of mRNA for the lack of lactose utilization. So here, if the RNA polymerase binds on, means the structural gene will be expressed. Structural gene will be transcribed into mRNA. If the RNA polymerase does not bind with the promoter, means it will not allow the RNA polymerase to bind, and that way the transcription will stop. So promoter is the region where it will allow the RNA polymerase binding, and if it is binds, means the transcription will take place. If it is not binding, means the transcription will stop. Then very next to that promoter region, there is one more region that is called as operator. So operator is nothing but it is acting somewhat like a switch. So if the switch is on, the transcription will take place. If the switch is off, means the transcription will not take place. How the switch is made into on and off, we will discuss it later. Immediately after the operon, there are 3 genes. They are named as Z, Y, A. So the Z gene, when that transcription takes place, means that gene will produce mRNA, which can take initiative for the synthesis of a particular enzyme. That enzyme is called as beta-galactosidase enzyme, which is one of the most important enzyme in this case, because in the presence of beta-galactosidase enzyme only, the lactose can be converted into the lysis of lactose, the breaking of lactose into glucose and galactose. Then the second gene, that is Y gene regulates the expression or the transcription of another enzyme. That enzyme is called as permease enzyme. And if this permease is present in the medium, in the bacteria, if that permease is produced within the bacteria, then only the bacteria can allow the permeability of lactose or the entry of lactose from the medium into the cytoplasm of the lactose. So even though you are adding lactose to the medium, the lactose from the medium, it should enter into permeability of the membrane of the E. coli, should allow or should increase, then only the lactose, more and more lactose can enter into, and then with the help of beta-galactosidase, the lactose can be converted into glucose and galactose. So the Y gene regulates the production of beta permease. And the third structural gene is A. That is, A gene will regulate the production of or the transcription of enzyme. That enzyme is called as transacetylase. The function of transacetylase is the transfer of acetyl group from one compound to another compound. But here in this case, direct involvement or direct role of transacetylase is unknown, even though it is found together. So whenever this promoter or operator is on, whenever the operator gene is on, it will allow the mRNA to bind with the promoter, and as a result, all these 3 structural genes will get activated. That is Z, Y, A. And as a result, they will transcribe into an mRNA. And from that mRNA, it is a polycistronic mRNA. That means single mRNA can transcribe more than one polypeptide. So here the Z region from the mRNA which is formed from the Z region will transcribe beta-galactosidase, helps in glucose, lactose to glucose and galactose. And Y region helps in production of more permease, which will increase the permeability. As a result, lactose will enter into the E. coli. Transacetylase, there is no direct connection, but even then that is together it is produced. So how this operator is made on and how this operator is made off. So here already I said, by the influence of I gene, by the activation of I gene which is constitutive in action, it will produce the it will undergo transcription, and as a result they will produce a repressor mRNA, And from this repressor mRNA, when it is translation takes place, they will produce a protein. That protein is called as repressor protein. So normally, this repressor protein will bind with operator and makes that switch off. The repressor protein, the figure it is given, the repressor protein will bind with the operator and makes the switch off. When this repressor operator complex, when they form, they will decrease the affinity of RNA polymerase to bind with the promoter. So that means the RNA polymerase cannot bind with the promoter. Once this binding is not taken place, means then the transcription of structural genes will not take place. That means the Z region will not transcribe, Y region also will not transcribe, and A region also will not transcribe. So the transcription does not take place. And in the absence of mRNA, that means mRNA production will not take place. Since the mRNA is not produced, the translation also will not take place. And as a result, in the absence of lactose only, I said. That means if the glucose is present in the medium. So how the lactose breaking enzyme, their production is not needed because enough and more glucose is present in the medium. They can directly utilize the glucose. No need of spending much energy for breaking the lactose or any other, because lactose is absent. Glucose is enough and more. The bacteria can live with glucose itself. The second one, how this system, so that condition is in the absence of inducer. So in the absence of lactose is no more needed because glucose is present. Glucose is the source of energy, best source of energy. But suppose the glucose is stopped or it is exhausted from the medium, then the bacteria will have to depend on some other source of carbon. So in that time, if you are adding lactose, glucose is absent, but you are adding lactose. So here the lactose itself, when you are adding, as soon as some amount of lactose will enter into it is permeable a little bit. So some amount of lactose will enter into the bacterial cytoplasm, and that lactose will act as an inducer. The substrate itself, here lactose only will have to convert into glucose and galactose. So the substrate, some amount of substrate will act as the inducer. So here there is a small isomerization takes place. The initial amount of lactose will convert into allolactose, an allosteric form only. So that allolactose will bind with the repressor and makes this repressor inactive, or else we can say makes this repressor incapable of binding to the operator. So the repressor inducer complex, in the presence of inducer, that means allolactose binds with the repressor and it will form a inducer repressor complex, which will make the switch on. That means the repressor will not be able to bind with the operator and the operator will be free. And once the operator is free from the repressor, then automatically it will allow the RNA polymerase to bind with the promoter. So once this binding of RNA polymerase to the promoter, then automatically the transcription unit will become active. Then this mRNA slowly moves from promoter to the structural gene. As it moves across the structural genes, that is Z, Y, A, the corresponding mRNA will be transcribed. And the lactose mRNA will be produced. And from that, the lactose mRNA will translate, and as a result, 3 individual enzymes will be produced. That is from the Z region, the beta galactoserase, Y region permease, and the A region transacetase. And this beta galactoserase will help in splitting or breaking lactose into glucose and galactose. And permease, because more and more lactose should enter into the cytoplasm from the medium. So the permeability will be increased so that more and more lactose can enter and as a result, the glucose will be produced by the breaking of lactose. And the bacteria can survive with the lactose. And then, suppose if the lactose in the medium is completely utilized, then how this lactose utilization has come to an end? That is, when the whole lactose what you have added is completely utilized by the bacteria. Then even the allolactose, which is acting as the inducer, also will be utilized by the bacteria. So that from the repressor inducer complex, the inducer is removed and it is utilized. So the repressor will be free from their action. Then this freed repressor will immediately go and bind with the operator and makes the operator off condition. That will decrease the affinity of RNA, which is already bound to the promoter, and as a result, detachment of RNA polymerase from the promoter, and the transcription unit will become automatically inactivated. And in that way, the transcription of lactose mRNA will stop. And from the transcribed, since the mRNA is not formed, the translation also will come to an end. So this is the process of switching on and switching off of genes according to the requirement of the cell. So this, apart from that, so when we read the concept of lacoperon was proposed by Jacob and Monod to summarize this one, and operon is the unit of prokaryotic gene regulation because operon is working in prokaryotes. Then gene regulatory unit which regulate the utilization of lactose in E. coli is called as lacoperon. The lacoperon consists of one regulatory gene that is called as I gene. I stands for inhibitor, and three structural genes Z, Y, and A. The I gene codes for a repressor protein which can bind with the operator and makes the switch off. Then the promoter or P site. P stands for the promoter. It is the site where RNA polymerase binds for the transcription, binds, and the initiation of the transcription can take place. And operator, which is the region next to the promoter, which is acting as a switch. And when the regulator repressor binds, the switch will be made off. In the absence of repressor, the switch will be made on in the case of lacoperon. Then the lactose, or another term, allolactose. Lactose or allolactose can act as the inducer, because the binding of inducer or allolactose only makes this repressor, with the repressor makes this inducer repressor complex, and that makes this, or that inhibits the binding of repressor to the operator. So it is acting as an inducer. Then gene Z codes for beta galactosidase, gene Y codes for permease, and gene A codes for acetyl transacetylase. So these are the functions already we have discussed only. Z, beta galactosidase, Y, that is galactose permease, and A, that is thiogalactoside transacetylase. And galactosidase will help in breaking down lactose into glucose and galactose. Then permease will increase the permeability of the lactose into the cell, and transacetylase, the function was not known. The more in detail, or to summarize the role of lactose inducer. So inducer means the component or the substrate which binds with the repressor. So here it is lactose, or another term exactly when we are saying means allolactose is the substrate for the enzyme beta galactosidase, and it regulates the switching on and off of the operon. Hence it is termed as the inducer, which will induce the transcription. The chemical which induces the transcription is called as inducer. In the absence of glucose, if lactose is provided in the growth medium of the bacteria, the lactose is transported into the cells through the action of permease, and lactose is broken down to glucose and galactose in the presence of beta galactosidase, and then can be used as a source of energy. So for that, the first initial amount of lactose is converted into allolactose, which is called as inducer, that will bind with the repressor and makes that operator on. Then repressor. So repressor of the operon is synthesized from the Ig, and the repressor proteins binds to the operator region of the operon and prevents RNA polymerase from transcribing the operon. In the absence of glucose, that means the lactose utilizing enzymes will not be produced because lactose is not provided in the medium. And not only that, one repressor is produced always, it is a constitutive gene. So always in the medium or within the bacteria, the repressor protein will be there that will bind with the operator, and it will make the switch always in off condition. And when it is made on means when the glucose is absent and lactose is added to the medium, that time the lactose initial amount is converted into allolactose, and then the repressor, it will bind with the repressor and forms an inducer repressor complex, which will not bind with the operator, and automatically operator is free from the repressor because repressor is already bound to the lactose. And the operator is on, and that allows the promoter to initiate the transcription. That means by the binding of RNA polymerase. So all these are the main points in this. Then when we move into, so this is the way the genes are regulated. So in that way, either the genes will be made on or genes will be made off in the transcription unit. Then when we discuss about the types of regulation, there are two types of regulation. One is called as negative regulation. The other one is called as positive regulation. So here lac operon is a typical example. Lac operon with the help of allolactose or with the help of inducer, that is a typical example for negative regulation. So what is negative regulation? In negative regulation, an inhibitor or a repressor is present in the cell. So always the transcription is inhibited with the help of a repressor protein, or in the presence of a repressor, then that type of regulation or binding of inhibitor or repressor prevents or suppresses the transcription of a particular gene, then that type of regulation is called as negative regulation. So lac operon is a typical example for the functioning of lac operon is a typical example for negative regulation with the help of repressor protein. The same way, there is a positive regulation also. So what do you mean by positive regulation? In positive regulation, an inducer molecule activates the promoter. That means in the presence of lactose. So here in the negative regulation, in the presence of repressor, the transcription is inhibited. So positive regulation means in the presence of inducer, the presence of lactose, the promoter, the presence of lactose, that means in the presence of inducer, the molecule activates the promoter, which promotes the transcription of mRNA, So here, positive regulation of lac operon is in the presence of glucose. Okay, glucose when it is binds, or if more glucose is present, means that will activate a certain, it is not given in your textbook because in the absence of or in the deficiency of cyclic AMP, in the presence of glucose, there will be deficiency of cyclic AMP, and that will stop the lac operon or the transcription of lactose utilization. So the lactose is present that, in the presence of glucose, glucose itself is only have to be acted as the source of carbon, because the lactose too broken down into glucose and galactose, energy wise the energy requirement is more because lactose has to be converted into glucose and galactose. So energy requirement, because their enzyme has to be produced, so a lot of energy has to be produced from transcription, then into translation, then only that metabolic pathway will take place. So instead of that, if glucose is there enough and more in the medium, then in order to avoid the wastage of lactose metabolic energy production, enzyme productions and all, they will automatically will stop the lac operon function. So that is taking place in the presence of glucose. So the presence of glucose can inhibit the lac operon, and then it is called as, or that will result in, or that can stop the utilization of transcription of mRNA for the lactose, and as a result the translation and all can take place. That is another topic only. It is not that kind of reaction action we need not learn. For the 12th standard in higher classes, we how this positive regulation, that is with the help of a certain enzymes and all, cap proteins, catabolic activator proteins and all, we'll say they will bind with the cyclic AMP, and then they will inhibit the functioning of lac operon. Then again, two types of negative regulated system, and the negative regulated or lac operon itself, or the operon itself, we can say there are two types of operons, that is inducible operon and repressible operon. So inducible operon, typical example is lac operon. So here the transcription is induced in the presence of an inducer. So a negatively regulated system may be either inducible or a repressible. So here the substrate itself will act as the inducer, and that the substrate means lactose is converted into glucose and galactose. So the lactose is the substrate. So the substrate, in the presence of substrate, that transcription is activated, and hence that is called as, or in the presence of inducer, the operon is activated, hence that type is called as inducible operon. There is another term we will use, repressible operon. Typical example is tryptophan operon. It is a feedback control. That means the end product itself. So tryptophan will be converted or will be produced as the end product. So that a product is regulating the further production of that metabolic pathway, metabolic product, then that is called as repressible operon. So there can be inducible operon. Typical example is lac operon. Then there can be repressible operon. Typical example is tryptophan operon. So tryptophan will be produced, and if it is in excess, then that tryptophan itself will regulate the production of, or will stop the further production of tryptophan. So it is that also not for you to study. So these are the main points how the gene regulation is taking place. So with only one simple example only, we have discussed this one. And in eukaryotes means this regulation will be even more highly complicated, because here in eukaryotes it is monocystron. Each and every polypeptide is in turn is regulated by a single mRNA, and that single mRNA is regulated by another transcription unit. And particular transcription unit, here we have learned inducer, then repressor, then modifier, silencer, enhancer, like that many other genes also will be there, and it is not in a continuous sequence. It can be located in different chromosomes in different regions. So that is more complicated only, and it is not, for us, we need not discuss much. So once again, lac operon is a gene regulatory unit which is regulating the utilization of lactose within the E. coli. And with the help of operon unit, which consists of inhibitor, which produces the repressor or I gene, then promoter gene, then operator gene, then structural genes such as Z, Y, A. And the presence of repressor, or the repressor always will be there. The repressor will binds with the operator, and they makes the switch off, so that the promoter cannot bind with RNA polymerase and the transcription cannot take place. Structural genes will be in the non-expressed condition. So no mRNA production, no protein production. But whenever in the presence of lactose, lactose will act as the allolactose, which will bind with the repressor, inducer, which will act as inducer. Inducer will bind with repressor. Repressor inducer complex will form so that the operator will be free from the repressor binding and the switch is made on. When the switch is on, the promoter will allow the binding of RNA polymerase, and as a result, the transcription of structural genes will take place. So ZYA will produce mRNA for the lactose. Then from that mRNA, three enzymes will be produced: beta-galactosidase, permease, and transcriptase. Permease will increase the permeability and more lactose will be added, and that lactose will be broken down into glucose and galactose in the presence of beta-galactosidase, so that the glucose utilization is lactose utilization is taking place, and finally it will be converted into glucose. And when the lactose is stopped, supply is stopped, then the inducer also will be completely utilized. So the repressor will be free and the repressor can go and bind with the operator. Then the transcription, the promoter will become inactive, or will be RNA polymerase will detach from the promoter. The transcription unit will become non-expressed. So this type of regulation is called as gene regulation. With this, we'll come to an end. Thank you very much.